Meiosis, we say, has the potential to produce genetic diversity in the gametes. And what we mean by that is an individual inherits chromosomes from his mother and his father. Those chromosomes are going to contain information relating to the same genes, but they can have different versions of the alleles at that gene. If that's the case, then it's almost like you have a deck of cards. You can shuffle that deck of cards and you can deal out a new hand of 23 cards. And that hand of 23 cards could be a blend of what you got from your mom in the ovum and your dad, what you got in the sperm cell. So, in other words, crossing over allows you to take the genetic information obtained from your parents and shuffle it in a unique way so that your gametes can contain a combination of mom's and dad's DNA on a particular chromosome. There are two ways to generate this genetic diversity. Crossing over is being shown in this one. The second way is via what's called independent assortment, and we'll talk about that in a later figure. So let's notice what we've got. We have a pair of homologous chromosomes. So remember back to the karyotype. Homologous chromosomes, we said, are similar in size, centromere location, and the genes that they possess. So we have three genes being represented here: the A gene, the B gene, and the C gene. My guess is at some point in your education, you've talked about Mendelian genetics. And in Mendelian genetics, genes or alleles of genes are represented using letters. And you have probably heard that uppercase letters represent what are called dominant alleles, and lowercase letters represent what are called recessive alleles. Now, we'll get into dominant recessive later on, but the idea is this: an uppercase C allele is different than a lowercase c allele. So we're saying that the homologue from one parent, we could say blue represents dad, has three alleles of the gene: the A gene, the B gene, and the C gene, and all of its alleles for each of the three genes are uppercase or dominant alleles, whereas the chromosome obtained from mom has the same information about genes. It's the A, B, and C gene, but she has three different alleles. She has the lowercase or recessive alleles. Now, there's nothing that says a chromosome can only have dominant alleles or recessive alleles. There are situations where chromosomes are going to have a combination of these, right? So don't get locked into that example. These are sister chromatids. So remember, when the DNA replicates, it produces an identical copy. So that means the sister chromatids will have the same alleles as the original chromosome before duplication. So each of these chromosomes has replicated. We now have a pair of replicated or duplicated homologous chromosomes. What happens in prophase of the first meiotic division, or prophase 1, is homologous chromosomes pair. In mitosis, this doesn't typically happen. In mitosis, the chromosomes line up end to end. But in meiosis, the first meiotic division, pairs of homologous chromosomes will align next to each other and what's called synapse, or attach to each other. When they're in that position, what can happen is something called crossing over, where information from one chromatid is exchanged with information of the other homolog member, their chromosomes. So, in other words, in terms of the color representations, we're saying what will happen is, instead of just having a blue alone, you could produce a blue with some red. Instead of just having a red alone, you could produce red with the blue. Now, a couple other things. Crossing over does not only occur at the one end of a chromosome. Crossing over can occur multiple times in a chromosome. It doesn't tend to occur in the region of the centromere because of the nature of the DNA in that region. But there's nothing that says it only has to happen once and only at the ends of the chromosomes. So that's just shown this is just shown as an example. I don't want you to think this is, the written rule as to how crossing over occurs. So if crossing over does occur, then notice what happens with respect to the alleles. Instead of this chromatid having only dominant alleles, it has two dominant and a recessive. And this chromatid, instead of having only recessive alleles, has two recessive and a dominant. So you see, what we've done is this process of meiosis has created genetic diversity, because during meiosis 2, when the sister chromatids attach, That means for this particular homolog, some of them will have what was regarded as the parental arrangement, and that's also called non-recombinant because you didn't get anything new from what was present initially. Dominant, dominant, dominant. So that's non-recombinant. And over here, recessive, recessive, recessive. Again, that's non-recombinant. But what you can get are recombinant chromatids. In other words, dominant, dominant, recessive, 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 dominant. So that results in genetic diversity in the gametes. In other words, a gamete will have a containing one of these two chromatids will have a combination of the individual's mom's and dad's chromosomes in a gamete from that individual. Please note this: crossing over only produces diversity if diversity is present to begin with. In other words, if both members of the homologous pair all had uppercase A, B, C on all chromatids, then you would have crossing over, but the crossing over would not be detected at a genetic level because you've swapped apples for apples. In other words, if I had crossing over, but this one had a dominant C and this one was a dominant C, you would have actual exchange of DNA between the non-sister chromatids, but it would result in no genetic diversity or no observable recombination. So this mechanism of crossing over has the potential of contributing to genetic diversity of the gametes of parents. And this is important because you think about if you have the same parents, some students are like, well, if I have the same parents, I'm getting the same genetics every time in a gamete. No. Crossing over can generate diversity so that a gamete from the father that fertilized an egg in the first pregnancy could be very different genetically from the DNA content of another sperm that fertilized at a second pregnancy.